to that link, please, in your browser. Do that now, okay? That's the most important thing actually you are doing during the workshop, because this gives you a link to all the materials we are going to cover here and more. So when I do that, I'm landing in here. So you can go via Gleb's email, or if you want to go the hard way, type in downloads.openmicroscopy.org. That's enough. Then you click yourself through the presentations into the right folder, which it's ordered logically. There is a folder 2021, and under that is a subfolder called GBI. Once you do that, then you land in this folder, and here are the materials. So from our perspective, as Gleb already mentioned, we are giving two courses. Pardon me. We are giving two courses, one today with two parts. This is, as Gleb mentioned, 1.5 hour plus 1.5 hour. And both of those are Omero courses. So that's why you will concentrate today on this walkthrough, which is the GBI Omero 2021 walkthrough for both parts of today. And you will also have the presentation with which I will start now. And this presentation is slightly confusingly, sorry for that, hidden in the Omero-workshop folder. If I click on that folder, you see that in fact it's containing a Reveal.js presentation. And you can follow the presentation on your browser as it is. It's not PowerPoint, you don't have to download anything, everything is there. Let's start, ladies and gentlemen. So today, we would like in the presentation to tell you what is OME. This is Open Microscopy Environment. What is Omero, and then give you some workshop overview. The Open Microscopy Environment is a group of people who are developing open source free software, and it purports to be a hub in the wheel, like the centerpiece of parts of the biological imaging world, but more and more other imaging worlds, connecting the bits which were originally actually not meant to work with each other, such as the acquisition software, which produces the raw data images, and data management, quantitative analysis. You would like, of course, to store the process data in a reasonable manner, close to your original data, view the images, and do some data management. One word for quantitative analysis. We don't do quantitative analysis ourselves, but the idea here is to enable the access to the data to the other third-party tools, which are much more advanced in analysis. Omero is a server with clients. Now, client is maybe a little bit cloudy word. What is a server? Start first there. Server is typically a machine which is remote and which has to be accessed somehow via virtually. Physical access is usually not granted to a server. It's a very powerful machine, and I'm sure that you heard that and you are using that in your institution. Now, Omero is mainly a server which is accessed via internet. As I said, I also don't have a physical access to the server which we will be using today. We will all be accessing it via internet, and the way to access that is you have an application on your local computer, and this application we call client, and this application will connect to Omero via internet. Now, the Omero server is containing all the image data which we will use today. We support the data on the Omero server are stored in their original format, and we are supporting more than 150 different formats. And this is achieved by the usage of the Java library called bioformats, which I'm sure you have heard already about. The data include different modalities. We talk about 5D image, which is the 3 normal dimensions, time and channels. And we have whole slide image formats, high throughput screening data, but also medical and general data. The Omero server is connected to a database, and the database stores metadata in a reasonable manner in a relation with the original data. We will go through that plentifully during the workshop. Just bear in mind that for us, metadata are regions of interest and segmentation, which you can add as a result of your analysis. You can perform search on the Omero server. And we have a flexible structure called Omero Tables, which is meant to be storing the complex analytical results which come from your secondary analysis. As I mentioned, we support high throughput screening data in a screen plate well format. These are the imaging formats from whole slide imaging, typically very large images. This is not a microscopy image, obviously. This is a CT scan of a whole brain of a human. The popular imaging format called DICOM. Now, as I mentioned several times already, you can analyze the data stored in Omero, and to achieve that, you would like to, and other things, you would like to use the Omero API, very probably. API stands for Application Programming Interface. And this is to be imagined if you don't have too much to do with computing. This is to be imagined as kind of a plug, an interface which you use to connect to the Omero server and access the data there. It's a very clever plug, and we have several of them. And the thing you are plugging into Omero is probably some script or program written in Python, Java, MATLAB, R, C++. And to document the same concept in the next slide, these are softwares, third-party softwares, which are very good in analysis of images, statistics, etc. And it looks like something different from the previous slide. That is not so. You can hopefully appreciate that each of those softwares is written in a programming language, such as Fiji is written in Java. Hopefully you realize that. And because we have that plug, this Java API, we made Fiji to talk to Omero directly. So are there some other bridges which we wrote ourselves, or the developers of the softwares in questions wrote helpfully themselves. And we will see some examples later. The main environment from the application perspective, the client-side application perspective, which we support nowadays, is a web-based environment called Omero Web. So you will spend most of the time in the workshop in your browser using the Omero Web client. And this one is an extensible. The extensibility is a property of Omero, quite pronounced. And you have the open with feature there, so that the plugins, so other bits of program, can be written freely for Omero Web, as it was done in this case. And you can open one image in all those plugins, which is quite nice. Now a little bit switch of the topic here. This is an Omero in use, and the use is for IDR. IDR will be mentioned tomorrow. Actually, I don't have to go into that too deep, so hold your breath. But the point to take home now is, please remember IDR technically stands for Image Data Resource. We will talk in length tomorrow. Technically, IDR is nothing else than Omero, okay? Now, all the bits and pieces which we will be talking through today are captured in Omero guides. It's a very good idea to use Omero guides on their own strengths. They are written in such a way that they can stand alone also without this workshop. And you can simply go to the presentation, click on the link, and here you go. That's how Omero guides look like. And I will be plentifully referring to these pages during the workshop. You, ladies and gentlemen, are typically facility managers. You would like to know how to present Omero in your institutions and for your users. So what should OME user know? Well, you probably would like to do routine training and user inductions, and you would like to attract users to Omero, okay? For that, we have bits and pieces, again, in the Omero guides. So you can go to the Facility Manager guide. This is for yourself. And to understand what the bread and butter user would like and need, there is a subsection in there. And what would you like to point the user to is, again, a much more simple walkthrough, which will simply show how to start using Omero. So all the links are here. Feel free to explore them. And without further ado, to the schema of today's workshop. This is again the same slide I'm showing over and over. Here is the Omero server, which is remote. All the data are pre-imported there for you. I will talk about it in the next slide. The server is sitting here in Dundee, UK. I'm sitting also in Dundee, UK, but I'm not sitting at the server. You hopefully realize that this is flat. This is a lockdown situation in Scotland, and the server is at the University of Dundee compound down the hill. Okay, so we are all remote to the server. The remoteness is highlighted again by the light blue line. Very good. Now on our desktops, there is certainly a browser running. Firefox and Chrome are highlighted, but you can use Safari, you can use even Edge. We are supporting many browser types. And inside is the Omero Web client, which will connect to the server. So this will be kind of a bread and butter of today's workshop. Then I will show some workflows with Fiji and Omero plugin for Fiji, which is running also on my Mac here in my room. And again, connecting to the server. And if the time permits, we will go into some analysis tools, which are in another environment. Again, on the cloud, remote. That's why the blue line is here. It's not locally. And we can, let's say, show how to run a CellProfiler on the cloud connecting to the Omero server. Again, if time permits, yeah, and if, of course, interest from your side is expressed. Now, I was highlighting that all the data are pre-imported to the server. By data, I mean, at this stage, mainly images. I'm not gonna lose the time with showing you how to do that in a demo. I have this slide to highlight that the import is done by a desktop application called Omero Insight. And that's what you would typically do as a first step. If you click on the right icon in Omero Insight, there is a widget where I will browse my local Mac or whatever machine I have. It works on Windows perfectly well and Linux as well. and select some images, put them into the queue, and then click import button. Not the rocket science. And after that, the images are imported. You can read it in the import guide if you click here, or we have some import video on YouTube. Let's draw your attention to that. Okay, the import video is inside an open microscopy channel. Okay, so if you want to see more videos, you can go find open microscopy channel on YouTube. Okay, open microscopy channel. The video I'm talking about is here, the third one. People start asking about such videos more and more. So we are trying to, when time permits, of course, to expand this portfolio. Again, here, this is another link to import guides. So feel free to explore those links. There are some more advanced import types, and this will be mainly the one which I would like to highlight is the in-place import. So you can be in a situation like with those 3 or 4 images I have on my laptop, and then you copy those images in the first step of the import onto the Omero server in full. So this is, of course, needing the same storage capacity on the server side as it was taking on the PC, yeah, or laptop. And after that, there is some other import steps, but this is not what I want to go into. So this is the classical import I showed in the previous slide. Now the in-place import is thinking in the following way. You don't want to double the capacity of the storage which is needed for your data after import to Omero. That's why you just teach Omero where the data are. And in this way, you import in place, that's how we call it, and Omero creates just a link to the images which are sitting on some storage place. One thing to remark here, the storage place is, of course, in this moment hugely important because the images are nowhere else. And if you somehow confuse Omero, by that I mean if you, for example, edit the names of the images or change the hierarchies here in the storage place, then Omero wouldn't see that. But otherwise, this thing is very useful for your situations where you have really vast amounts of data already on some other server, like Share in your institution, and you don't want to, or you even cannot duplicate the data amount by importing them into Omero in a classical way. We are open source organization, but of course we are not developing the software for free, and the money is coming from grant funders. Our main funders are Wellcome Trust and BBSRC. So thank you very much for them, for the support. And now I can see there is a question. Yeah, and how much data space is allocated to a user? Okay, regarding in-place import, can I have several different storage server linked from Omero? So the second answer is yes. To Ofra Golani, regarding in-place import, can I have several different remote storage server linked from Omero? Yes, you can. You simply mount them on the machine where the Omero server is installed. Is there a limit in data size that can be used? No, there is no limit. And how much space is allocated to a user? Okay, this is a question which shows that the whole ecosystem is not completely understood. So let me make sure that we understand each other here. How much space is allocated to a user is completely up to you. What do you mean by you in this case? You, I mean your institution, namely, mainly. So the idea here is that Omero server is a software, okay? And that's what you get for free. But every software needs some hardware to run on. The hardware is not what you get for free. The hardware is something you have to provide on your side. download that free software onto that hardware and run it. This implies that everything you store, the storage capacity, the volumes, etc., must be also on your side mounted to that hardware. So Omero is for free, but you will not run it without costs, because you have to pay for the storage of the data. This is still up to you. And typically what we have in mind is that the institution will have one Omero server, the institutional Omero server, and that's where they will install the free software called Omero, and that's how it's running. Okay, so so the hardware maintenance, backups, upgrades, security patches of the server, etc. This is all up to you, and you must do it. Okay, and the OME team is just delivering the free open source software. Okay, and you are running the show basically in your institution. Then you probably see from that how much space you allocate to every single user in your institution is completely your choice, your call. And how you handle it is very institution specific. So I don't want to even go into that unless you want to discuss later. Okay, I hope that I answered the questions. Well, there are some more questions. Well, Jean-Marie will feel free to interrupt me if more input is needed or something to be shown, and I will simply go to.
Yeah, move forward. I will answer the question in the chat. So
thank you. I will simply go to the walkthrough. Okay, so let's double back on myself because I'm losing the thread. Sorry for the clicking now. I'm closing the tabs here and just trying to make order. So don't get disturbed. It will take a second. I will go back where I was, and this is the first link I was pointing you to. Yeah, ladies and gentlemen, are you there? I will leave a couple of seconds for you to find your footing, because I myself had to have them. So this shows that this is necessary. And what are we concentrating on is the third item here, GBI Omero 2021 walkthrough PDF. Yes, you got it, ladies and gentlemen. If not, then tell me or scream, because that is your lifeline. Okay. I'm opening that in a new tab, and that's what you should be seeing. Okay, it starts GBI 2021 Omero workshop. The presentation and PDF version of the workshop are available at Then there is a summary. So we will go through Omero core concepts, data management, and metadata. Okay, for that, we are now switching. I showed you a couple of slides, how to import data. We're discussing about that. And now we want to switch to Omero Web. Why? Because Omero Web is our main client. That's where we are channeling our efforts, and that's where we are putting new features on. That's where all the nice plugins are in. How to get to Omero Web? Well, first of all, there is an email which was sent helpfully by Gleb just a couple of minutes before the start of the workshop. Why do I log in as Train 1 actually? Because Train 1 has bigger portfolio of data. In Train 1 imported more data. Okay, and so I have here a tree on the left-hand side, which consists of projects and datasets. These are containers in Omero. We have two levels of containers, and a project, this is the blue thing, can contain datasets, which are the green things, and the datasets then contain images. Okay. The dataset might be under project, but it might be also without a project. The image must be under a dataset. We have then some other structures for the screenplay 12 format, but let's not go into it unless there are questions. After the import into Omero, I have here some images. So these are the CT scans of the whole brain, side by side with some classical microscopy images with the convolution images next to them. These are whole slide image formats, in this case SVS. If I click on a thumbnail in the center pane of an image, I can see the metadata. First of all, in the general pane, in the right-hand side, there are the most important metadata, such as the owner of the image, the acquisition and import dates, pixel type, and the number and names of channels. You can see that this image is something like 100,000 by 50,000 pixels. So big images, not a problem. Good. And to make sure that we understand again the principle here, all the images are imported and stored in Omero in their original format already mentioned. And how is it possible that all these images are side by side in one kind when they are in their original different formats? So this is a DV format here. And as I said, this is a DICOM format. Well, this is possible because we have the bioformats library, which reads the images on the fly. Okay, so if I would be to open, let's say, this image in full viewer with double clicking on that, the bioformats would be called. It would take the appropriate reader and read the image for me on the fly. Okay, so there is no conversion step to another format in Omero. This might change in some future, but now, as we see in Omero 5.6, there is no such thing, and we are storing the original data. Okay. Now let's go into the acquisition pane. There are some metadata which were read from the original file during the import. Now the import is also deploying bioformats, and bioformats are reading all the items of metadata which are stored in the original file itself. Of course, if it's in the file, then it reads it. If it's not there, then unsurprisingly, it cannot read that. And displays it in this in this manner, which might not be the most user-friendly. But for that, we have a model, which means we know kind of, let's say, what a numerical aperture of the microscope is. And then we would take such item from the original metadata and put it into the database. And in the database of Omero, we can display it from there in such a manner as I'm showing right here, which is much more user-friendly and much more easy to read. Okay, very good. Then you have a preview tab on the right-hand pane, and that's where you do rendering settings. Rendering settings are basically the way you display the image on the screen. Changing rendering settings does not change the original image. In fact, we never change the original image. Okay, you can imagine that Omero is creating a lot of artificial objects above the original image, and your original image is kind of cocooned and protected there. And anytime you would like to grab it, it's there for you. You can download it and have it back in exactly the same way you imported it. Okay, but these rendering settings are very useful, and you can think about them as a pair of goggles which you are using to view the world. You can change the bars here. This changes the min and max displayed value. This is probably not something surprising. As I'm doing on this image, and then when I'm very drastic and click save, you will say that these settings will be saved on that image, and the thumbnail will be regenerated and shown. Very good. Now, if I go back and imagine an experiment which might have been done by a scientist in this data set, such as I have stained for a protein of interest in a green channel, and I would like to see if my staining worked or not. For this, I would like to, let's say, switch off all the channels except for green. This hopefully makes sense to you. Then I would like to adjust the rendering settings in the green channel. Something like this. And I would press save. Well, that would be for one image, but what if I use this save to all button? Let's try it. All here means all the images inside the dataset. Okay, but this is my dataset with the whole staining, and in one click, I put the same rendering settings on the whole dataset. So if I then go here above the center pane, I can filter the images further. Many scientists are encoding the metadata into the names of the images. So if I, let's say, type scram, these are the images which I think should be stained. Yes, some are stained, some are not, but this is already my experiment. So yeah, I have some information from that. And then when I go and filter for in, I have another batch of images. This should be my controls. And indeed, I can see that the controls are clean. So that looks good for me. Okay. So in this simple manner, I have verified that my experiment worked or half worked or whatever was the scientific outcome. I didn't have to worry about whether the rendering settings are comparable. Omero made sure that for me. If I want to go back to the rendering settings, which were there before, I can go to the left-hand pane, right-click on the dataset itself, go to rendering settings, and say set imported and save. And there is a short warning dialog, and then I'm back in the status which it was in when we started to work with this data set. And now to show you some cooperation aspects here, we have here some thumbnails.
Petr, sorry to
Yes.
interrupt. There was a question, can you show how to add multiple data set to the same project? So that's a question in the chat. So
Very
it
nice.
would be easier to show
Yes.
it than talking through it.
Thank you very much. First of all, I can create a new project like so, and I say this is a GBI project. Okay, and did you see here is an icon? This is the most natural. Now I'd like to add more data sets, so they can be new, and I can click on data and type dataset 1. So I clicked on the icon, and because the project was selected, the dataset is automatically added there. Another one is dataset 2. Okay, very good. But if I want to work with existing data sets, I can simply drag and drop. The Fiji data set used to be an orphan, but I just dropped it in my GBI project. You can also import the data sets straight into the project. So that would be in the import stage. There are simple user interface widgets to achieve that goal. So it's not a rocket science. If I want to remove that data set from the project, I can use this cut link, which will cut the link between the GBI project and Fiji dataset, and that will pop the Fiji dataset free, and we call it so-called orphan. So orphan means that this dataset now doesn't have a father project. Okay. Very good.
And sorry for Darren, which indicated it's colorblind. That color, one of our colleagues, Josh, created because that's something we need to address. We've not, for people colorblind, the difference is not great. So sorry about that, Darren.
Sorry about that. Yes, of course, we will have to work on that. Okay, if there are no questions urgently about this one, let me come back to the small thumbnails. On the bottom right, if you select an image in your siRNA HeLa dataset in the central pane, go and click on a preview. And on the bottom right, there are some thumbnails of the rendering settings, which are done by my colleagues. Who are my colleagues? Well, this is you. In this case, you are the other colleagues in my group called Lab 1. I can select the thumbnail of my colleague Barbara McClintock. You might think that you are not Barbara McClintock, but almost certainly one of you is Barbara McClintock. This can be either a gentleman or a lady. Of course, there is no telling. We didn't care about that. And the thing is that Barbara McClintock is simply a user in Omero, which is in the same group and is able to view my images. Who is me now? I'm trainer one, and it's written who am I is written on the top right as usually is. So my first name is trainer one and my second name is trainer one. Okay, that's a bit silly, but you see that Barbara McClintock has much nicer name. And I can now overtake Barbara McClintock's rendering settings on my image by clicking on this small thumbnail and clicking on save. Okay, this changes and I have overtaken her images. This changes also this small thumbnail of trainer one. So you can see that on the same image, Barbara McClintock can have her pair of goggles and I have still my own pair of goggles. Now I simply decided to reuse Barbara McClintock's one and I click on save to all. Okay. And that's how Barbara McClintock would like to see my images. Okay, very good. Barbara McClintock has her own portfolio of her own images, but this is a duplicate. And if she changes her rendering settings on that, I can't see that. On this image, we all have, let's say 50 instances of the same images which are independent of each other. And by the way, please don't ever delete anything on this server, okay? I can go back, maybe I'm not happy with the Barbara McClintock's way to view my images. I want to view it my way. So I go again and set imported and save. Very good. Now let's go to the full viewer. Let's say if I take an image which has many Z planes, such as this one, it has 40 Z sections. Okay, I can double click on the thumbnail and the plugin for Omero Web called Omero iViewer opens. This is our main full viewer at the moment. In the iViewer are the thumbnails of the images. These are the other images from the same dataset. And I have here a Z slider for going through the Z sections, okay? And I can have also a T slider if this is a time lapse. This is not a time lapse. The rendering settings are here repeated. That's fine. You can also draw regions of interest in Omero iViewer. There are already some regions of interest which are associated to planes 15 and 23. So first of all, when I click on those regions of interest, which I have there, I can jump between the planes. The viewer is jumping as well. That's very nice. But you would maybe like to know how to draw a new region of interest in the first place because your image might have none of that. And this brings me to something I forgot. So really apologies. Let's go back to the web client. And in the future, also, I will be using this knowledge of yours, which I hope to impart on you right now. On the top left, there is the lab one, and this is the group which we are members of. In order to see each other's images in that group, and this is a read annotate type of group, read annotate means that we can see and annotate each other's images. If I click on that, a drop-down menu goes down, and I can see here the list of all members, and I can see Ronald Fisher, portfolio of images. This means on the top right, it says trainer one. This is because I am still trainer one. I'm logged in as trainer one, but I'm not viewing data of trainer one. I'm viewing data of Ronald Fisher, which is signified in top left. And in this way, you can view any data of any member, such as data of trainer one. Okay. So now I'm going back to my own portfolio and this will happen in a second. And okay, and I'm coming back to this new tab, which I opened from the SIRNAI, HILA, and I will show you how to draw regions of interest. Sorry for the sidekick, the regions of interest in iViewer. So first of all, let's draw a simple rectangle. This might be a little bit different than you are used to from other softwares. I click, then you would try to drag, but this would just move the image. So my finger is not in use. I'm having a mouse here. I'm holding it in the air. I'm just moving the mouse once I clicked. Then I drag the mouse. Okay, my fingers are in the air. I just drag the mouse. And then now I click again, and the region of interest popped up here is the blue one. Okay, on the Z-plane number 18. I can disassociate the ROI from the Z-plane by clicking off that lock here. Let's leave it alone now. And now let's draw some freehand polygon. Freehand polygon is a little bit different story. I will, with my left hand, press Shift on my keyboard and then click and drag. Click and drag means the same way you are used to from ImageJ, let's say. Okay, now I can't show you my right hand or my finger from right hand because it's in use. Okay, the mouse button is down. Now I let it go and I let go the shift, and again a polygon has appeared here in the list, and this is highlighted now like so. Okay, so hopefully you understood how to draw a freehand polygon. This was a little bit different than to draw the rectangle. And then I can do, first of all, I will save the regions of interest. Then I will select them. Then I will go to ROIS and export as table CSV. This is a rudimentary image analysis, which you can get from Omero. We already said, I will open it directly with Microsoft Excel. And I have here an image. It has 12 lines. Okay, the results of the image have 12 lines. This is because I have 4 regions of interest. Okay, and each region of interest is an image which has 3 channels on. The image has 4 channels, but only 3 channels were on. Okay. The being on and off is regulated here in the settings. You can see that the 4th channel is off. Okay, so that's why there are 12 lines in the output, and I can see that yes, for the for all of them is area. If I would have drawn a line, then there would be rather a length and some simple statistics. But very interestingly, the points which define the regions of interest are also exported in this manner. Okay, so you can get them from there. So much for drawing of regions of interest. Now let me show you another feature. If I would like to see the split view, which is in many viewers a mainstay, I just dragged and dropped on the free place the image of the same thumbnail, and a new viewer opened. Okay, so I'm here correctly in the settings pane, and I would like to achieve a split view, which means that I would like to have a different channel in each of those viewers of the same image. Okay, so this will be my blue channel. This will be my green channel. And now I have the split view. Okay, just adjust it a little bit, but you can see that that they are not synchronized. Okay, so I'm moving in the blue channel now, but the green channel is doing nothing. I can change it by putting both of those viewers into the same synchronization groups. On the top left, there is a lock icon, and I can lock it in the number 1 group as well as this one. When I do the first action, such as scroll through Z, and or zoom in and out, you can see that it's, as it's written, Z and T are synchronized and view is synchronized. This is meant by view being synchronized. So if I pan or zoom in, the images are synchronized, and this is meant by Z and T synchronized by dragging the bar here. Now, to get you into the routine, every drag and drop into the free pane opens a new viewer. Okay, that's fine. every single click on another image on the left will change the view in the active viewer window into that image, such as so. Okay, single click it was. But if I double click on yet another image, a new image will open. So drag and drop into the free pane and double click will open a new viewer, whereas single click will just change the view, or I can drag and drop into an existing viewer. Okay, and this will also just change the view like so. Okay, so this is viewing in iViewer and drawing regions of interest. And
Peter, could you show maybe the lock unlock next to ZOT, because there were questions about propagation. I've just replied to Leandro. At the moment, we do not have a mechanism in the web of, you say, I draw on the middle Z and propagate to 5 above and 5 below. We do not have this propagation yet in place. It's available into our desktop client that Peter show a slide for import. You can do that, but not in the web. The only thing you can do in the web at the moment, if you decide not to attach to a given plane, that's what Peter is going to show. So that's a feature currently missing in the web viewer for that.
So you can see that when I'm now moving through the Z, okay, this region of interest is kind of everywhere. This is because it's not attached to any plane. I achieved that by unlocking that log on the Z. There is a script which is in the main web client, where you can export batch ROI export, and it will give you some data on that, but it will not give you a full joy on those unassociated regions either. So it's a work in progress, yeah. But that's how to you can achieve something by unlocking that log, yeah. Feel free to explore, please. Okay, any more questions? Otherwise, I will go back to the web client. I think we plentifully explored iViewer. And there is, of course, a link to the guide in your walkthrough. And let's go back to I call that the web client. Yeah, this is the Omero Web interface where we started our journey from. And let me draw your attention to what happens when I select again my favorite data set called siRNA HeLa. And the reason it's my favorite is because it's richly annotated. And I'm now talking not about the annotations which are in the acquisition pane, which are basically coming from the original file. I'm talking about the so-called secondary annotations, which you can add post hoc after import, or even during import into Omero. I'm going to use those annotations here. I already saw that I can filter by name when I drop down the menu above the center pane. Above the thumbnails, I will now choose to filter by tag. Okay, I can choose tag, anaphase. I have several images in anaphase. How is this achieved? Well, very humbly, the images were recognized as anaphase by our colleague and actually author of these images, Will Moore. And we simply went ahead and added the anaphase tag on them. Like so, we clicked on the plus and we created a new tag. Be mindful, don't create a ton of new tags, but of course explore at will. And then we popped it into the right-hand pane and saved. You can select and tag one or more images in one go. And you can remove the tag like so. There is a remove tag button over here. Very good. Now I can select and choose another tag to filter for, let's say metaphase. Again, you can see it has the metaphase tag. And that's nice. And there is a pro-metaphase tag in the same vein. Yeah, I don't want to lose your time with that. But if I now go and select by rating, I can see that I have 3 images here which were annotated in the manner I just started to highlight. This means that, for example, this image, because it's a metaphase image, First was with its peers tagged with a metaphase tag. Once that was done, this image was highlighted as the best out of the metaphases, and it was tagged further, it was annotated further by adding a rating. Okay, and this is a 4-star rating. And so it was done, this is the best one of the prometaphase, etc. Okay, so you can see now the annotations in action. This is for tags and ratings. This is all nice, but our massively, like miles ahead, most popular annotation type is key value pairs. So let's concentrate on the key value pairs here for a second. So I can, for example, have a protocol lab book of a scientist where unknowingly the scientist is creating key value pairs. It's always not just one thing, like it's a metaphase, that's not enough. Typically the protocol goes temperature was 37, incubation was 17 minutes. The solving agent was methanol, etc. So this is always key and value, key and value, key and value. This captures much more information, and the information is also much more better organized, as well as it's searchable. So for example, you can see that I have here key animal and value rabbit. And if I go to the search and I search for animal rabbit here with the colon denotation as highlighted here, this is animal colon rabbit. Okay, I will find just that image. And this says that it must have key value pair. The key must be animal. The value must be rabbit. Okay, like so. If I now change the search, this is a big global search. Okay, it doesn't search just in the data set. It searches on the whole server according to the widgets which are here. For me, it searches for all groups and just for my data, but you can search for all groups for all users' data. This means for all users, you have the right to see the data of. Okay, now if I remove the rabbit and the colon and search for animal, 2 images pop up. This is basically saying, find me any images which have a term animal somewhere in their metadata. Now this is animal rabbit, okay, in the key value pairs, but the animal could have been anywhere else as well. And this is animal goat, okay? So if I want to look for animal goat only, then you can see this already says, as you can guess, just find the key value pairs. The key must be animal, the value must be goat, okay? And indeed, I'm finding this image, animal goat, okay? Very good. And I can now go back to the main view by clicking this browse button. So here is my animal goat, very good. And I can multi-select the images like so. I'm having 4 images. And if I do here some typing, like let's say GBI, okay, and value is great, okay. So this is my new key value pair, slightly unorthodox, and is attached to all 4 images in one go, okay. I can remove if the key value pair is the same. I can remove it in the same way. So I'm using command click here on my Mac. You can see that the GBI is great, is a nice statement, but it's ultimately not that great. So I will remove it from all 4 of those images. Okay. So that's how to work with key value pairs. The reason I'm really dwelling on that is that even IDR, which we will see tomorrow, is really insisting on having key value pairs, as well as many users say, yeah, this is the ultimate annotation for us. Nevertheless, let's not underestimate the attachments. The attachments in the next harmonica are giving you the opportunity to attach anything. you like, any file you like within reason of size, of course, to an image or more, or to a dataset or project in Omero. How to do that? You select an object. I am not saying an image. You can select indeed a dataset or several of them, and click on the plus, and then you can browse the local hierarchy here. Okay, that's brilliant. I have the measurements here. I can say open and then accept, and you can see that the new attachment will pop up here in a second. And these are the measurements I just created in front of you from the other image in Omero iViewer, and they are now attached. They are on the server. Now you can upload a MATLAB file. Why not? You can upload an Elastic project. Yes, you can. But don't expect Omero out of the box to be able to read MATLAB file or expect Omero to read Elastic project. No, Omero will not do that. Nevertheless, the file is stored there in the right place on the server for you for later peruse. This is number one. And number two, in the analysis part, we might not have time to go into that, but you can harvest such, let's say, MATLAB file, simply connecting your local MATLAB to Omero and teach MATLAB how to find that file and then harvest it as a recipe. And or Fiji, we can talk about macro in Fiji. Let's say you pop it on your data set and then tell Fiji via a script, okay, the macro is there, download it via API, use it, insert it into the script, execute it on the whole data set, stuff like that. Okay. Furthermore, Omero will do something nice if the file is PDF, CSV, or a text file. It will, in the big search inside the text of such files. Okay, so that's some bonus for you from out of the box Omero. Well, and of course I can download the files back from Omero like so. I clicked on the attachment and say open in Excel, and it's reopening here. It's of course the same file I was creating already for you. Very good. This would be the annotations. And let's now quickly go to show you Omero figure. Okay. So once you prepared your ground like this, you would like to annotate and analyze. We will do the analysis after the lunch. Okay. Once you have your analysis, you would like to publish. So I can select an image, let's say from a data set. From here, IDR 0021, I can select one or even two, and then open them by right-clicking. This is the open with, and click on Omero figure. Yes? This opens the images in Omero figure. What is Omero figure? Omero Figure is a plugin for Omero Web, which allows you quickly to create publication figures. Okay, let's not think directly about sending that to nature. You would like to show it to your colleagues, to your boss, to collaborator, etc. And this will quickly allow you to create a figure where you can communicate a lot of information based on your images. It has a connection obviously to Omero, and on the flies asks Omero about the metadata associated with the images which are captured in the panels inside a figure. This spares you the classical ordeal of a scientist who has to export images as, let's say, JPEGs or PNGs from the software of interest, let's say size Zen. Okay, nothing against size Zen, of course, but once you export that, you have to export the concrete Z plane, and then you go to Adobe Illustrator and find that there was a mistake, and go back to Zen and find another Z plane, the loss of time already first 20 or 30 minutes, okay? The Omero figure allows you to double back on yourself at any time and quickly create much more effective workflows. with much faster results. So I selected both of those images. I will align them to grid. I will make them yet smaller. Okay. Then I do copy paste. I simply do the classical way, Control C, Control V. Okay, couple of times. Let's say 3 times. Again, adjust. I can nudge with the keys on my keyboard. I can make them a little bit bigger. Now, the objects inside, these are centrioles, inside those images are quite small, so I can zoom them in by selecting all and zoom them in. Okay, so this is too much. I can go back on myself a little bit. Very good. That the lower side, lower part is central. That's fine, but I'm not happy with the top one. So I select the top row, and this is how I like it. Okay, so I adjust that. Then I would like to change the rendering settings. So this will be my blue channel. This will be my green channel, and this will be my merge. Okay. Then I will start adding labels. So first of all, let's start with the scale bar because the image is calibrated in Omero. I can add a scale bar like so. If not, then the pixel size is clickable and I can add the scale bar right here like so. I click on that and then retype the number. Okay, that's good. Now I checked the label, but I know that the image is quite small actually now. Okay, so I have to go for 1. I hit enter. 1 micron is quite right. I can change even from micron here to other units. Very good. So this is the scale button. Now on the top, I would like to have the description of the channels. How do I do it? In the labels tab, I can add channels, and I can add them to the top. I can make them a little bit bigger, like let's say 14 or let's say even 18, and then say add and watch what happens here at the top. Okay, these are the channel names. Where are the channel names coming from? Well, from Omero, obviously. These are still Omero images. And Omero figure just asked Omero, what is the channel name of that image I'm using in this panel? And Omero answered and delivered the answer. Okay, very good. So on the left, I have another feature. I would like to add the dataset name because in this case, the protein stain 4 is encoded in the name of the dataset. So I will add them to the left vertical. 18 is very good. And you can see that this image is in multiple datasets. So this is up to me to sort it out. And this image is in the CDK dataset. Very good. So I'm actually kind of done, except I would like to make a insert of this one. So I can copy the image, okay? And I can now delete the labels on this one. And I can now go to preview, zoom in like so, okay? And this would be my insert, okay? Like a crop. There is a specific crop button, but I don't have to use it. I can simply now directly go and sort out the zooming of my Firefox. Okay, very good. Sorry. So there is a copy button over here, which was hidden a second ago. Sorry about that. I can copy that region from here and paste it in another tab right here, like so. Paste. And that's where my crop is coming from. Now I have it. Okay, I can still change even the properties to put it as yellow. That's fine. That's the crop. I can nevertheless also add regions of interest from Omero, which were there from my previous analysis. Let me just zoom into the figure again so that you can see it better. I'm here on the insert and I would like to have the segmentation region around the region which was done in Fiji in a way which we will see after the break. Okay, so the Fiji simply created some segmentation on that image, and I'm now adding it by clicking on the edit in the labels tab. I say load rows. Okay, that's the one I like. Okay, I add it there. So this row is coming all the way from Fiji, which harvested the image from Omero, did the segmentation in Fiji, saved the region of interest on the proper image back to Omero. Then we went to Omero figure and harvested that region of interest and putting it into our publication. Okay, very good. So this is this panel, and I would like now to go and add some more images to the same region, to the same figure. This is done by selecting a couple of images like this one and this one, and then click on the link and copy, and then go to the figure and add image. I paste that link. The images are here. By the way, why didn't I go with open with? Well, because it would open the images in a new figure. That's not what I want. I want to add them to an existing figure. Okay, so I select them too. Again, snap them to grid very quickly because we are running out of time. Sorry for that a little bit. And copy, paste. Yeah, and then again the same routine: blue channel, green channel, and this is a merge. Okay. And now again, I would do the channel labels on the top. That's quite fine. Actually, 18 is a suggestion. Yes, but it's a wrong position. So I say the top, and it's not dataset name, it's channels. And go for me. Again, these are the channel names. And on the left, I can add something. I can add the key value pairs. Okay, key value pairs. On the left vertical, and I add them, and it says antibody. Antibody would be as good as anything, I think. Include key and a label. Let's see how it does. Antibody goat, yeah, that's fine. So this one is the antibody goat one. Okay, of course I can have another key value pair over here if I wish so. And so on. Of course, scale bar, label, add, done. Very good. So this is my figure. Now I can save it. This is the GBI figure. This figure is now saved in Omero, and but this doesn't somehow duplicate the data or images or anything like that. It's just a lightweight recipe how to recreate that figure when I come back to the figure later. And you can see that that's how to achieve that. I went file open, and then I have a list of figures which were created previously. And of course, my new GBI figure is in the list. I know that there is a cooperation aspect here as well. I can go to my colleagues trainer 1 or Omero Api figure 2, which I know that is quite nice. Open it, and here you have an example of what everything you can achieve in Omero figure. You can have quite large images. Again, the insert, this is the 100,000 pixel by 50,000 pixel image. You have time lapses here. Now, if you watch what will happen with the time lapse, if I zoom in, it says 42 minutes in the label. The label nevertheless is alive. And if I click on the preview and then go in the time, watch what will happen with the label. Now it's at 61 because the plane is different and the metadata about the delta T were fetched on the fly from Omero, and my labels are changed like so. I can now save the copy of this figure of my colleague trainer 2 into my portfolio, etc., etc. But also I would like to export the figure and show them to people which don't use Omero, or send them to publication. It will create a PDF for me on the server, and then I will be allowed to download such figure. Okay, if you bear with me a second. And very good. And I will open it with preview. Now the PDF My broadband is not best in the world. The PDF is now going locally to my Mac, and voila, I have my figure. It has 2 pages, but my PDF has 3 pages. Why is that? Well, because some metadata of the figure are here. So first of all, link to the figure itself is here. So if I click on that and I'm logged into Omero, the figure will appear immediately for me. So 2 things, I had to be logged into Omero and I had to have a right to see the figure. By right, I mean within the Omero permissions system, okay? And you can send such link to your colleagues as well inside email. That's also quite useful. You can grab that link actually from the URL. Then later, when you send the figure into a journal, typically after a very long time, the journal comes back and says that the figure in panel 7 has some mistakes and the panel 7 has to be exchanged. Nevertheless, you forgot what original data did you put into panel 7. But here are all the links to the images in Omero. If you click on that in the PDF, that's precisely the image which sits in the panel 7. And you have now the links and identifier inside Omero. Okay, I thank you for your attention. I think I'm a little bit over time.